share with you today what we do after an ROV dive. During the dive, we've requested different samples to be collected at different stages. These could be biological samples, geological samples, or water samples. We can sample using a push core, Niskin bottles, using the manipulator arm to place a specimen into one of the bio boxes, or we can take a suction sample. After the ROV is recovered and safely secured back on deck, then the ROV pilots will give us the go-ahead to go out and collect our samples.
So once the ROV is on deck, I just grab my helmet, I grab my life jacket and also a long line so that I can secure myself to the ROV. When it comes to suction sampling, I know that each sample will be in a different chamber, so I just pick up very carefully. Sometimes we use suction sampling for very sensitive animals. So we just take the chamber with us and we take it to the lab and we process it in the lab. And for not so sensitive samples, we just put them in a black box. So for example, rocks or other corals that we know that will be used for stable isotopes, for example, we just put them in the black box and we just put them in a bucket with water, and then we also process it in the cold lab.
Each sample is photographed next to a ruler so we can get the exact size of the sample, and each sample has its individual label. On this label, there's information such as the cruise number, the dive number, and the sample number, so we can match every single sample back to the original remotely operated vehicle footage. Samples might be placed in the -80 freezer, put into the dehydrator oven to dry out, or simply stored in ethanol.
The samples that we collect during each dive, sometimes we use it for different purposes. So sometimes we collect samples to be analyzed in terms of stable isotopes and fatty acids. Sometimes we take samples for taxonomy. Sometimes we just take samples for geochemical studies, and then we just store them accordingly. So for example, for stable isotopes, we just freeze them at -80 degrees. Sometimes for taxonomy, we either dry them or just store them in ethanol, and that's what we do with the samples.
After each dive, we also make sure to update our spreadsheet, which keeps track of all the samples.
So it's quite important that we do a list of all the samples collected, because sometimes we're dealing with protected species. So what I do after each dive, I go to OFF and I see the moment in which sample was collected, like the LEDs and LON information, and also the time and the depth. And then I just put this data into a spreadsheet so that I have all the information organized there.
Once you've done this, you're done for the night. You can head to bed and be ready for the next ROV dive in the morning.